From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on May 9th, 2024. Hello, Micronauts, and welcome once again to today's Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today, believe it or not, is the 40th episode of Matters Microbial. I am just amazed. I should think about doing something special for the 50th episode, don't you think? Those of you who watch and/or listen and spread the good microbial word to others, many, many thanks, and keep it up. First, a friend kindly sent me some of this glass from my Wunderkammer. The fluorescence under UV light is due to uranium in the glass. It was quite a popular thing early in the 20th century, and you know how I feel about things that glow. I put a link in the show notes so you can learn more. The semester at the University of Puget Sound is now over. As is my habit, I took my research students, I call them Doc Martians, a name former student and currently fabulous bioinformatician Kim Dill McFarland first coined, to lunch. Here is my lab group that do research with me during the academic year. I gave each of them a glow-in-the-dark Doc Martians pin by Hartiful as a thank you, as you can see. Two of my Doc Martians graduated this year, and I'm very proud of them. Melissa poses with her favorite enormous giant microbe, chlamydia. She hopes to become a physician. Samantha, on the other hand, poses with her enormous HPV plush. She'll be applying to bioinformatics PhD programs next year. Now, no student is ever obligated to get me a thank you gift, but they did. A tardigrade nightlight that they each signed. It was a wonderful semester and a wonderful lunch. I am very, very lucky. Now, our guest today, Dr. Joe Handelsman, has been a friend and mentor to me for many years, endlessly patient. And it was from Joe that I first learned about ice nucleation by Pseudomonas syringae that I demonstrate every time I teach microbiology. The bacterium makes an ice nucleation protein that promotes ice formation in very cold, pure water. It never fails to amaze students, especially when it is thought that most snowflakes have such a bacterium at their center. It is also how man-made snow is created. The freeze-dried bacteria are called snowmax. I'll put a link to this wonderful phenomenon and its role in nature in the show notes. Also, Joe and her colleagues created the Tiny Earth student antibiotic crowdsourcing program that I've mentioned on this podcast before. Students finding new bacterial strains creating antibiotics will become a key tool to battle multiply resistant bacteria. This amazing cure-based program has certainly influenced how I teach. As you can see, even my first-year students can isolate bacteria that appear to have antimicrobial effects on a series of relatives of pathogens and then identify the organism in question. It has really helped my teaching and inspired so many students. Again, I will put links in the show notes. Not only that, but Joe Handelsman recently wrote a book, A World Without Soil, the Past, Present, and Precarious Future of the Earth Beneath Our Feet, that illustrates how vital and threatened soil has become. Many take soil for granted. After reading this book, you will never call it dirt again. As Joe exclaims, soil rocks. One of the things that most interests me in microbiology remains microbe-microbe interactions. We are all far too focused on single isolates and single colonies. But how to study such things? Joe and her colleagues have identified a simple system of 3 microbes working together that are a model system for complex interactions in the areas around roots, the rhizosphere. Thus, today you will hear about THOR, the hitchhikers of the rhizosphere, and zorbs, mobile biofilms, among other fascinating topics. Formally, Jo Handelsman wears many more hats than I do. She's been a professor of plant pathology and microbiology at the University of Wisconsin at Madison, officer in the American Society for Microbiology, a book author, been a professor at Yale University, worked in the Obama White House to promote science, and is currently director of the Wisconsin Institute for Discovery. And I am just beyond delighted and honored to welcome her to this podcast. Welcome to our Quality Quorum today, Jo. It's great to see you.
Thank you, Mark. Great to see you too.
Well, I got to tell you, it's wonderful to have you here, and there's so much to talk about. And I wanted to tell you that it feels almost like I'm repeating myself whenever I'm speaking to you, because so much of what you do and think about are things that I introduce in the classroom. So I wanted to thank you for that, of course. But I think a good place to start is many people don't appreciate how complex the soil actually is. And in doing so, we can move from that to the issue of THOR and then maybe mobile biofilms. Can you tell our listeners and viewers why they should love soil the way we do?
Well, that's, of course, one of my favorite questions. And soil is important for so many things that happen on Earth. Everyone thinks of the food supply that comes mostly 95% from our soil directly. But there's so much more to soil. It serves as the largest water filter on Earth. It serves as the largest repository for carbon on terrestrial Earth, more than all the plants and trees combined, and 3 times as much as we have in the atmosphere. So in terms of climate change, soil is critical for carbon balance. And it's the source of most of the antibiotics that we use in medicine. So soil is pretty important for a lot of things that people just don't recognize it for.
You know, it's interesting to me. There have been microbiologists in my past who've taught entire courses where they take a pinch of soil and they isolate a wide variety of microbes from them. And in that sense, it reminds me a lot about folks who study the gut microbiome. There are so many different organisms. They're all interconnected. There are battles for dominance. There are associations of mutual benefit and everything in between. And it's so hard to study a super complex system. And as I said at the beginning, I think that humans are often reductionist and they look at just one thing under one condition. And maybe that's how we start how to study things, but it doesn't tell us anything about the complexities. So would you mind talking a little bit about this simple system? Well, I shouldn't use the word simple, should I? This system of 3 organisms working together.
I think that we study, as humans, we study things both at the most complex level. We've always done that, looking at the earth as a whole, looking at soil as a whole, looking at the human body as a whole. And then we have this impulse to isolate all the pieces. And the 20th century was filled with science that focused on individual pieces. And that's how we sorted out so many of the secrets of biology. And of course, microbiology has depended upon pure culture for 150 years, and it's been a fruitful 150 years. But studying the nature of communities in pure culture, a single organism, just doesn't make any sense. A community is obviously an interaction of organisms. And so what we struggled with was, how do we study communities without all that complexity that you were referring to of the human gut or soil, where there are literally thousands of species living together, but still work in a community. And that's when the idea of THOR came up. We wondered if we could come up with a system that represented the major groups, but didn't have every representative of those groups. And so our aim was to have 3 members of the community, because 2 is a binary interaction, and more than 3 sounded way too complicated for starters. So we started with 3, and that's how we came up with THOR. And we started with an organism that I've studied for a long time, Bacillus cereus, and then we built on it from there.
Now, it's interesting because the Bacillus cereus that you use was something that I used a lot in my classes for years and years, and it's a really wonderful tractable system. I don't know, by the way, if you know how different it looks on low nutrient media compared to high nutrient media. It completely changes its morphology of the colonies. It is the coolest thing ever. And, it's the kind of thing I was all excited about. And then I talked to other people and they said, yeah, we've seen that too. And it makes story of my life. But it's really interesting because here's this one organism. And the interesting thing about Bacillus cereus to me, as a gram positive, it has this super, super, super thick layer of peptidoglycan. I'm always trying to be silly and maybe a little flippant. And so the peptidoglycan is like a net that helps protect bacteria against changes in osmotica. So you might think of them almost, oh, please forgive me, like Spanx for microbes. But the fact of the matter is that you have at least 50 layers of this stuff. That's just Bacillus cereus. And that is where I first noticed some of the stuff that you were discovering, which brings us to the refrigerator.
We were studying Bacillus cereus strains from roots of plants, soybeans and alfalfa in the field. So this was going out into actual Wisconsin soybean and alfalfa fields. And we would isolate Bacillus cereus, which is very easy to do based on colony morphology, as long as you use the same media. As you said, it changes with the type of media. And we would pick the colonies and streak them out for single colonies and then colony purify again. And we thought we had beautiful, pure cultures like good microbiologists would. And then we would put a whole array of them because we had this collection of about 500 we were aiming for to store away. And we would put those plates in the refrigerator. And we'd come back in about 3 weeks. And all of a sudden, there'd be not just these pure, beautiful, creamy white Bacillus cereus, but there'd be these blebs of yellow stuff, usually yellow, growing out of the Bacillus colonies. And we didn't know what that was. At first, I always said, oh, it's just contamination. You just don't have sterile good technique. And so for a long time, I dismissed it, but my students kept seeing it one after another. And so by the third student, I said, okay, maybe this is something consistent that we should be looking at. Maybe this is just not contamination. And the fact that so many of them were bright yellow suggested to me that there was a pattern here. And as scientists, we can't ignore patterns. And so eventually I had a very brave student who calculated the frequency at which these other organisms appear. And it was consistent across field seasons and across soybean plants and across alfalfa as well. About 5% of the Bacillus cereus isolates would have these blebs. And that also sounded like a pattern to me. That could not be random. And so we started purifying the blebbing things, the yellow and related colonies coming out of the bacillus. And we realized that there was a great pattern. 80% of them fell into the phylum of Bacteroidetes. And most of them were Flavobacterium and Cytophaga, the gliding, beautiful yellow gliding bacteria. And then about 20% of them was they were a mishmash of other species, and other phyla were represented, mostly Pseudomonas, but some others as well. And we found that among the Bacteroidetes members, the Flavobacteria, they had this funny characteristic that they wouldn't grow in root exudate. Now, these were all isolated from roots. So you'd think, okay, they grow on roots. But in fact, they didn't grow on root exudate by itself. When we added Bacillus cereus to the root exudate, the Flavobacteria grew just fine. Then we began to study, well, what's the factor in bacillus? And we found that it was just the peptidoglycan. So as bacillus grows and it sporulates, it flips off fragments of peptidoglycan from its cell wall. And it turns out those fragments are fantastic as a carbon source for Flavobacteria. And so if you feed the Flavobacteria with peptidoglycan in the root exudate, they grow just fine. And so that seemed to be what the bacillus was providing. And so that began to suggest that this was a very, very deliberate, evolved symbiosis where the flavobacteria had somehow attached themselves to the bacillus by a mechanism we still don't know, and was using the bacillus peptidoglycan as food as they lived and grew on the alfalfa and soybean roots.
For listeners and viewers, it's important to know that when you played out dilutions or streak to single colonies, the idea is the colonies are derived from one particular cell as they divide. So the idea of isolating anything from a mixture usually takes three different single colony isolations. But what we're hearing here, and this is something that I suspect is actually very common, maybe not in model systems that we use in the laboratory, but any surface can be colonized by bacteria. So is it a surprise that some bacteria can colonize the surface of other bacteria, and therefore they can kind of be carried along as you go through all of this? And I'm with you 100%, Joe, is if you look at something very strange that doesn't make sense, your first move is to discard it. Very similar to some of the stuff that Von Cooper, Mike Travisano, and Rich Lenski have looked at with Pseudomonas syringae throwing off these different morphs, smooth and wrinkled. And your first look at that plate is, well, it's a contaminated plate, but it's not. And so this is a fascinating idea that takes me back to the idea of microbe-microbe interactions.
Absolutely. And, we weren't the only ones who saw this and thought it was contamination. I don't think I've ever given a talk on this where someone hasn't come up to me afterwards and said, usually whispering it, we find that too. And we always thought it was contamination. And one person even said that when they went back to their freezer stocks from, decades of accumulated strains and did a 16S analysis, they found that 80% of them contained some other bacterium. And these were all stocks of organisms isolated from the environment. As you say, the model systems, E. coli, is typically pure in the lab. It's been in the lab for so long. But some of these direct isolates from natural environments are definitely bringing their hitchhikers along.
And I think it's a lovely expression. I myself, of course, and I'm sorry, you've heard me say this a bunch of times, want to know if they're burrowing into the peptidoglycan, as I suspect they are. But we do know because there's a lovely illustration from one of your earlier papers where you put peptidoglycan in the media, and the flavobacters are making areas of clearing because they're using the peptidoglycan itself, which is modified sugars and short peptides. So it's not a surprise that it would be yummy to many microbes.
That's absolutely right. It's a great food source, and so it's a very typical microbial interaction. What is a waste product for one microbe becomes the food for another one.
Do you remember, Joe, the business with fragments of peptidoglycan acting as a signal for symbiotic and pathogenic interactions? The Bordetella pertussis toxin, TCT, actually is a segment of peptidoglycan. On the other hand, Vibrio fischeri kind of sloughs off peptidoglycan, and that acts as a signal to the squid. So this idea of what could be food for one could be signals for another.
Yeah, food or signals or something else that we haven't discovered yet. And peptidoglycan is complicated enough that it has some real specificity as a signal, and that makes sense. It's not just some long polymer that the flavobacterium can chop up and eat.
I have to tell you, Joe, parenthetically, that we find lots of flavos in water bottles, just telling you.
Your water bottle experiments scare me, Mark.
Why? We found antibiotic producers in them.
Oh, well, then they're okay,
Yeah,
right?
there you go. It's just an environment to be colonized, and you're going to be selecting for things that are able to bind themselves to a surface. That's the idea. And it's not a surprise that they're going to be duking. I shouldn't be using martial terminology, but it is true there are antagonistic and mutualistic associations taking place.
Absolutely. And antibiotics play roles in both. Antibiotics can be signals or they can be warfare molecules. Unfortunately, the dogma has always been that they're for warfare, and people talk about the arms race between microbes, and That's probably true in a few cases. Some cases, we don't know how many because it hasn't really been studied rigorously in more than a few systems. But there are certainly others where the antibiotics will never reach killing levels. They're just not toxic at the low levels they're produced. And those are the levels that are more likely to act as signals. And we have some evidence of that as well.
I think Julian Davies has written some really interesting stuff about that a few years ago. Uh,
Yeah, that's what really got us started. Julian wrote a beautiful paper about 25 years ago about the role of antibiotics as signals. And the first experiment he did that was really illuminating, that we've now repeated with more current technology and found very similar things, was that if you treat salmonella with sublethal levels of antibiotics, you find a vast change in gene expression. Many genes are either turned on or turned off. And that's exactly what we find in our model system of the three organisms, which, by the way, we call THOR, which is the hitchhikers of the rhizosphere, for where they came from. But it's the same pattern that one of the organisms produces this antibiotic that causes gene expression to alter in thousands of genes in the community.
Joe, one thing that our listeners and viewers might appreciate knowing is what is the role of these microbes on the roots of soybeans?
That's a really great question. We know in great detail what we can get one of them to do, which is Bacillus cereus protects soybean roots from disease by producing zwittermycin, which is an antibiotic that we discovered a long time ago that Bacillus produces. That inhibits the growth of oomycetes, which are eukaryotic pathogens of soybean roots. So we know that bacillus, when it's there, at least in sufficient numbers, can have that effect. We don't know for this particular Pseudomonas and Flavobacterium that we use in THOR whether they have a role in plant health or affect each other on the root. And we're moving toward root experiments with all of the members together, at least in the laboratory. So we're hoping to be able to answer that. It's a great question, and I wish we knew more.
So, in my mind, I have this cartoon of this ginormous Bacillus cereus and this skinny Flavobacterium grabbing chunks of the surface and munching it as it rides it. So that's the image I have in my mind. But there's a third member from time to time, which is a Pseudomonas. Is that true?
Yeah, we decided to have a community of three members. We needed another hitchhiker, so we picked one very typical Flavobacterium that acts as a hitchhiker, and then the third one is in that other 20%, the ones that are not in the Bacteroidetes group. And one of those was a Pseudomonas. And the way that we picked it was we looked at interactions among many of the hitchhikers, as well as just random isolates from roots that were not hitchhikers. And we asked which ones inhibited or were inhibited by other members. And we found that this Pseudomonas ended up at the top of the heap, that it was the most interactive of all the organisms we tested. It inhibited more of the rest of the community, and it also was inhibited by some of them. So we decided that that was just based on chemical communication. That might be a good place to start. And so that was a Pseudomonas coriensis, and we began to characterize that organism as the third member, and that's what became what we now call THOR.
I think what's really important to remember is that, and please forgive me for using this terminology, Joe. I refer to a lot of bacteria isolated from nature as being undomesticated. And by that, I mean there are a lot of tools that haven't been developed for their study. So I think what makes THOR stand out, in my opinion, is that you can manipulate every member of the association genetically to ask informed questions and see what phenotypic changes take place when you alter specific genes or groups of genes.
That's what excites us because it allows us to take what is truly a complex system and use reductionist tools such as genetics and biochemistry to study it. So the first thing we did was we sequenced all the genomes. And so THOR has about 17,000 genes among the three organisms. We developed genetic systems to be able to manipulate the genes in each of the members, or we borrowed systems from other people. And we developed ways of getting genes into each organism so that we could label them with fluorescent labels. And then we developed a metabolomics profile of the whole community and the individuals so that we could figure out which metabolites appeared to come from which members. So we have what we think are the key tools of microbiology that enable us to dissect the community using the community as the unit of study instead of an individual species.
Reminds me very much of the early days of studying biofilms in general. And people would look at one organism in a biofilm. And certainly I've taught it that way when we do simple lab exercises. But the fact of the matter is that's not how biofilms exist in nature. And there are many emergent properties that take place when you bring different organisms together that you couldn't necessarily predict. I mean, the level of interaction between these three is in your system is very remarkable to me.
Yeah, I find it phenomenal that there are so many types of interactions. You know, you mentioned earlier the morphology of Bacillus cereus. Well, one of the things Bacillus cereus can do is it spreads on plates and it forms these sort of dendritic growths out from the colony. That is enhanced by the other two members of THOR. To different degrees and in different ways, but both of them will induce it. We don't know what the benefit is to bacillus of those growths and of the spreading, but we definitely find it a repeatable phenomenon. And we even have mutants that don't induce the spreading or that of Bacillus cereus that won't spread. So yeah, that's just one of many interactions, and then the genetic interactions are truly phenomenal. The Pseudomonas coriensis produces an antibiotic that we discovered called coriansin. And it's a very simple molecule that vaguely looks like a signaling molecule, like a quorum sensing molecule. And it even has a long tail, just like ACL, just like AHLs. And what's remarkable is that thousands of genes in all three organisms will be up or down regulated by the presence or absence of coriansin. So when we use a mutant that lacks coriansin and replace the normal Pseudomonas coriensis with that mutant, the gene expression patterns change radically, and it's literally thousands of genes that change in their expression. One molecule doing that. And then you imagine all the other metabolites that are being exchanged, and it's like a volleyball system. They're just sending signals back and forth and inducing each other to change behavior.
I'm kind of seeing it more like a really complicated dodgeball thing going on
More
because
like
there's
that's better.
so many different things taking place back and forth. So, as I understand it, you've dissected out these three members, and when you mix them, does it recreate this? I hate the word, well, I don't hate the word hitchhiking, but because it's completely apt. But so can you recreate this phenomenon where they persist in Bacillus cereus after you recombine them?
We have not been able to replicate the hitchhiking phenomenon in the lab. And so it's interesting that we've only found the hitchhikers when we isolate Bacillus cereus from field grown roots. But we've never seen it, and we've looked at thousands and thousands of Bacillus cereus colonies from soil. We've never seen it in free growing bacillus from soil. So that says that it is unique to that, or at least specialized in that root environment. And what we may need to do is try to replicate that environment in the laboratory and maybe try soil grown roots in the lab. And it's not the kind of thing that's easy to get funding for. And so it's too much of a fishing trip. Can we replicate this phenomenon from the field in the lab? People don't really want to fund that. So someday perhaps we'll have the funding to be able to do that experiment. But it's kind of a big experiment because the frequency, at least in the field, is only 5%. So you have to isolate a lot of Bacillus cereus to get any kind of meaningful data.
Well, it's really interesting because you now have them marked. You now have them carrying drug resistances. So I'm not saying you can force an association. And by the way, I would worry about kind of in vitro evolution in that particular scenario. But I mean, you have all the parts to do some such fantastic work, and it's been such a pleasure to see from afar because I've watched it since the first days, and I've taught it with variations pretty much every year.
And you were the ones that named them hitchhikers. You deny that, I know, but we got that word from you, and we'll always be grateful because one, it gave a great image for people to understand the system. They are hitchhikers. They get a free ride with Bacillus, but it also was the H in Thor. It gave us the ability to give Thor its cool name.
Well, it's something that as you know, I need a German word, and the word is jealous and proud all crammed together because such a great system to look at, and my hands just quiver at ideas to try with that. But this is not a surprise for me because I'm interested in lots of the ways that organisms interact with one another, and I don't know if I showed you this before, but when we were testing for antibiosis with some of our isolates from soil and mosses and lichens, what we found is some of the escape relatives, Joe, looked very different on different backgrounds. So we would get dendritic looking colonies with one background but not another.
Oh, that's so interesting. I think
Yeah.
that's probably a more common phenomenon than we have thought about in the past. Because I think we've selected these very tamed organisms. They're, like you said, domesticated, and they form only one kind of colony, or we only culture them on one kind of media, and so we only see that one kind of colony. So there's an awful lot to do in just looking at the natural history of these organisms.
There's and I've said it before on the podcast, but there's a lovely quotation by the late Isaac Asimov, the science fiction writer, where he said that science doesn't progress, he's being silly, with some damp Greek emerging from his bathtub shouting Eureka. Yeah. Actually, science progresses when someone goes, that's weird. And that's true. When you see something unusual, and that's what I tell students all the time.
Yeah.
Just use your eyes, use your brain. And the machines that are expensive and cool and do wonderful stuff are important. But who operates those machines are probably the most important part.
Sure, the questions are the most important part of science is what are we curious about? What do we want to learn? But I have to say the machines are pretty cool these days. And just the idea that we can look at all the genes and their expression in a community of three organisms simultaneously, to me, is remarkable. It's the kind of thing I wanted to do in graduate school many decades ago, but the technology was so far from even imagining that it wasn't something that we could even talk about rationally. It was like a science fiction idea.
Even in the stuff that I do at my level, I can find unusual variants. And for about $75, if it's a known genome versus the quote evolved genome, I can find out what genes have changed.
Hmm. Yeah.
And that's something that I can bring to students that's just remarkable. And I hesitate to think what it'll be like in 15 or 20 more years.
Yes, I try to dream about it to imagine what it would be like, but I probably don't have the imagination to come up with the ideas that others will and that will change science once again.
So I think something else that might be fun to talk about is an emergent, I don't know if we can call it an emergent property, but I love Zorbs.
Mhm.
It's just something that you've talked about, and I'll put some links in the show notes. And when I call them mobile biofilms, the first thing that I want to remind the viewers and listeners is a lot of the gliding bacteria can move across surfaces very effectively. And so when I isolate them, say from water bottles or from the soil, if you're not careful, they'll cover a whole plate really quickly. Now, sometimes they use surfactants, like they make their own slip and slide essentially. Other times they have rotary motors on their surfaces, but this idea of gliding motility is associated a lot with surfaces. And you had some wonderful results in a recent publication on this particular topic with flavobacteria using a different way of moving around than we've seen before. Do you think you can tell us about it?
Well, sure. We were very surprised to find out in collaboration with an engineering lab. And this is another example where the technology enabled the science that we never would have discovered probably any other way. Our collaborators in the BB lab in engineering here at Wisconsin did this beautiful experiment where they looked at some of our bacteria under oil. And it turned out that in their system under oil, flavobacteria would form these very regular, very consistent globular structures. They were three-dimensional, they were spherical, and they moved on surfaces. And they were clearly a biofilm because they were held together in the middle by extracellular polysaccharides. We could stain them and see that. And they didn't roll on the surface. They were many, many cells put together, but they didn't roll like a ball. They slid across the surface. And it turned out that the cells at the bottom of the what we call the zorb, the spheres, would sort of act like walking cells across the surface. And they created sufficient force just by their movement to move the entire spherical structure across the surface. So we were stunned by that because biofilms are thought of as stationary structures. That's how we've always treated them. And there are very few examples of biofilms that move. And this is clearly a biofilm that moves actually quite rapidly across surfaces, about a micron a minute, which for a big globular structure of bacteria is pretty fast.
One question that occurs to me as I read your papers and look at the videos, things of that nature, is to ask the question, is this something that only takes place under oil, or do you think it's also something that's taking place on the surfaces of roots?
Well, we were worried about that for a long time. Is this just an under oil artifact? And it's interesting, even if only under oil, just because
Hmm.
it's such an organized process and seems so specific. But we found that we can actually transfer the sorbs to other environments like polystyrene plates, and they will exist there. And then I'll give you a tiny little sneak preview of something we haven't published yet, that the sorbs form and move in animals. And so we think that they're pretty relevant.
Well, that just made my afternoon. Thank you. I think it's remarkable. And again, this is something that one wouldn't have expected to see, but it's using a different approach. I give a lot of lip service on this particular podcast about different faces, different eyes, different ideas. But that's really what advances science. And this level of collaboration you're talking about is fascinating. And no one had expected this kind of thing is what I'm getting at.
We weren't
And I
looking for it for sure, and it was
right.
completely unexpected.
And I think sometimes, and I don't mean to be philosophical about it, if you're looking for a particular answer and your mind isn't open, that's all you see is that particular answer. You don't see what else could be going on. And with all due respect to Barbara McClintock, who wrote a very influential book to me, A Feeling for the Organism,
Oh, she
and
didn't
I do
write it.
think that
It was written about her. She didn't write
I'm
it.
sorry, you're correct. You're correct. She had some unusual things to say about the book when it came out. But yes, and I will put a link in the show notes to that fine book. I think it's by Keller, but I'm not sure.
Yeah, it is.
In any event, it is a fine book, and one of the things that comes up in the book is you get a feel for the organisms that you study. And anybody who's worked with a series of organisms for many years kind of gets a feel for it. But I do want to say you have to be ready for something that doesn't fit, that doesn't fit what you know, and be open to it. And that open mind is important. To me, that's like childlike wonder. And I'm completely unashamed about that because that's how I've always felt about science.
And
So go ahead.
not all scientists want to find the unexpected. And some are more open to it than others. And some systems certainly yield the unexpected more than others. And so I think we need all different kinds of science. But as you say, the kind that comes from a collaboration of people coming from different perspectives, different disciplines, with different biases of their own, can open doors that we never would do just with one scientist or one discipline.
So let me ask you, what's next for THOR?
Well, what we're working on now is, first of all, cosorbs. We found that the flavobacterium will not only form sorbs by itself, but it will encapsulate other bacteria, like the other two members of THOR, and many other bacteria, Staphylococcus, E. coli, lots of them, and carry them along. And in one really weird result, the cosorbs with the other bacteria move faster than the sorbs. Now, the other bacteria don't necessarily have to be motile themselves, so they're not contributing to the motility, but somehow the speed of the sorbs moving across the surface increases when they have a payload inside. So go figure, bacteria will come up with new surprises every day. So the looking at cosorbs is a big part of what we're doing now, and trying to figure out where and when they're important in the life of the bacteria. We're also looking at the role of other antibiotics produced by members of THOR, and we have one cluster of genes that's really interesting in Bacillus that is very different from known clusters for known antibiotics. So we're sure that it's a new molecule, and we're trying to isolate the molecule that is encoded in that machinery. But that's an interesting one because we have a hint that it might be acting as a signal to the bacillus itself. We're not sure how much we know it responds to other organisms, but we don't know if it is a signal to other organisms. So those are a couple of the places that we're going, and looking also at gene expression across many, many different habitats, many conditions, and with different combinations of organisms is one of our big areas. And in particular, I'm interested in how communities are invaded. So, what makes a community vulnerable to invasion? What makes an organism a good invader of a community? And we're hoping to identify the genes that are involved in that so we can get at the mechanisms of invasion. You know, that's a broad ecological principle. All communities of all types are invaded, and we have some pretty good ideas with things like invasive weeds or invasive insects, what characteristics make them invaders, very effective invaders. But we don't really have a lot of ideas for bacteria that have been tested rigorously and what makes them good invaders of a community. So that's what we're hoping we'll figure out.
Joe, you've mentioned thousands of genes among 3 different organisms that make up THOR, and trying to see how they're related and coming up with models. Do you think AI can help you come up with some approaches to this?
Yes, absolutely. AI has already been useful in looking at the patterns of gene expression and finding the consistency or lack thereof of certain kinds of genes going up and down. And in the future, we're hoping that AI will help us build predictive models for community behavior.
Oh, I think this is just wonderful. I can't begin to express it. It's because what I like again is the approach, which is large scale, but you're able to use dissection tools at the fine scale to ask questions about the large scale. And
The
and
way
and
of
that's
putting
the way it well, I think it's a good way to put it, and you can't always do it. If you were using approaches like this, say with the human gut microbiome, and there are people who can make modified changes bit by bit, it's much more complicated. And I'm guessing that you're going to see the same basic principles because nature tends to use the same strategies. Why not, right? They've had billions of years to develop them. And as you and I both know, first evolved, last extinct. That's what we say about the microbes, right?
True. And we also think that we can quantitatively build predictive models.
Yeah.
We're collaborating with statisticians and modelers who can quantitatively describe what the bacteria are doing and what's needed for invasion. Then we can test those models on other microbiomes, other bacterial communities. And maybe that's how we'll finally evolve some of the general principles of what works in all communities or most communities.
Oh, I think it's fabulous. Now, Joe, you've also, if I can switch gears as we start to slide toward the end of our session today, you've done a lot of work of representation in science as well, and I'm going to put a link up to your bias paper from a few years ago. Did you want to say a few things about this work of your own?
Yeah, it's an interesting parallel with some of the biases that we were talking about a few minutes ago in science. Scientists have dealt with the problem and the issues of bias for a long time. Like you said, if you're not looking for something, you may not see it. And if you're looking for something, you may well see it because there's a bias there. And as a result, scientists have used the concept of the blind experiment. We blind our samples. We don't put the explicit labels on them so that when we're taking spectrophotometer readings or we're counting colonies or whatever, we won't necessarily know what answer we're looking for. And so we've always believed in bias in science. And yet, when I first started talking with scientists and groups of scientists about the dangers of unconscious bias about people because of either their ethnicity or their gender, there was a scoffing and a very consistent denial in audiences where they said, we're trained to be objective, we're scientists, so of course we don't do that. And of course, one of the elements is these are unconscious biases. This is not a cognitive process. And that's, of course, what scientists are trained for is the cognitive process, the logical deductions and inferences. We're not talking about that. We're talking about things that come from deep in our unconscious. We don't know where they actually have originated. So that's the first misunderstanding. But the other is, wait, we use blind experiments all the time where the investigator doesn't know the nature of the treatments. So if we're so unbiased and we're so objective, why would we have to do that? So I knew that that argument didn't make sense, but because a lot of the unconscious bias literature was done by psychologists with either undergraduate classes of psychology students or people in the workforce or non scientists in academia, I couldn't say absolutely, nope, we have direct evidence that scientists do this as well on the human bias part, even though it seemed completely predictable given 50 years of research on human unconscious bias. But because I didn't have a direct answer for that, I just had a logical argument. I said to one of my postdocs and I were talking, and I said, what are we going to do about this? And we both kind of knew right away what the answer was. We had to do the experiment. And so that's the paper you were referring to that basically takes a description of a student, gives it, in one case, randomly the name Jennifer, in the other case, the name John. We sent the different labeled, we sent the different, we sent the description of the student with either the name Jennifer or John to researchers in chemistry, biology, physics departments across the country and major universities. And then we asked them a series of questions. And it turned out exactly the way all of the studies on unconscious bias had come out, that they were much more likely to hire John than Jennifer. They would pay him more. They would be more likely to mentor him. And they thought he was generally more competent. And these are identical descriptions. The only difference was the name on the description of the student. And so that, had a really big effect. I found it actually kind of embarrassing how much press the experiment got because my psychology buddies had been doing this for 50 years. They knew what the answer was going to be. One of my collaborators, Jack DeVideo, who really kind of was one of the originators of the unconscious bias field, and I were talking one day and I said, well, we might not find anything. And he just burst out laughing at me. Like, I've done this so many times. He said, I know how it's going to come out. And sure enough, it did. But because it was done with scientists, these objective people who are trained in logical objective thinking, it made a big flurry of press and people started noticing it.
It's an interesting thing because I remember when all this happened and you and I talked about it and you had to walk me through that it's not personal, it's not about choices, it's independent of that, it's much deeper. And that's something that's really hard to take, but you really can't argue with the data. And that's something I've really tried to live my life to do and let the data, because I have a saying, data don't care, because they don't, they just are. And there are lots of people that pick up a football and they turn it into politics and they run with it, and that's their thing, and that's cool. But what's interesting in this case is where there's genuine data about it that people can look at, and it's really hard to ignore it. And believe me, Joe, ever since reading that paper, it's been much on my mind.
Well, that's great to hear. It's certainly been on my mind. It made me much more cognizant of the dangers of bias in everything that we do that involves people.
Yeah, well, and that it may be just engraved in our genes. I'm not going to go all sociobiology on you, but maybe this idea of trying to forge associations and exclude people is something that's really hardwired. Call it primate politics, for lack of a better term. Maybe that's what we're dealing with there to some degree. But we have more of a frontal lobe that we can try and find ways to deal with that, I'm hoping. Although some days are better than others, I must admit.
I think we all struggle with that, being logical, cognitively based people and being influenced by unconscious things and emotions and all of that obviously is a challenge. But it's interesting that unconscious bias can be based even on differences between groups that actually exist, and we still get the wrong answer. And the experiment that I just love along those lines was scientists took uh, pictures of people standing in doorways, and they asked their participants how tall the person was likely to be. And they overestimated the heights of the men and they underestimated the heights of the women. So, yeah, there is a real difference. Men on average are taller than women, but it led them to the wrong answer, which to me is such a great example of even when there's a legitimate, statistically proven difference between groups, we judge the individual incorrectly because we're applying that general bias. And then think of the danger of when there is no difference between the groups and we just imagine there's a difference, then we're also applying that to individuals. And so I think it brings to mind the all of the dangers of applying expectations of a group that may or may not be true of the group to any individual. That's what we call prejudice, and that's wrong.
So this is a little bit philosophical here, if you don't mind, kind of microbial philosophy. So we talked a lot about unconscious bias, and I'm not going to say that microbes have cognition or they make decisions, but when you think about issues like THOR, there have to be mechanisms by which they're able to exclude or invite particular organisms. And do you think that there might be relevance to this idea that there might be some hardwired behaviors, for lack of a better term?
Yeah, it's a really interesting way to put it because I think that is the nature of invasion or resistance to invasion. So, how does a community either invite a member a new member to join or keep it from joining? And how does an organism recognize a community that it wants to join, if you don't mind the anthropomorphism? My only problem with is extrapolating that kind of group behavior to human behavior in a sort of sociobiological way that justifies certain human behaviors, because we have decided not to follow the natural world in many areas. And I think it's really important that we don't justify biases or group exclusion based on what the natural world does.
No, I couldn't agree more. I mean, this is one of the things that I see happen when researchers talk about microbial behaviors where there are aspects of cheating.
Mhm.
And then people tend to transfer that idea. Well, probably you've heard me say this. Do you know the business with Pseudomonas syringae where it forms a mat?
Mhm.
So there's that one morph that forms a mat. And then within that mat, you will get mutants that don't expend any energy on making the cellulose. And they're
Mhm.
able to grow more quickly because of that. And so the mat sinks. And then I know it's just me being silly, but I say we've all had experiences like that. And while that's true for group work, perhaps in a student situation, because all the students just groan that they've been through that themselves, it is important to remember that we're talking about another set of, and I hate saying perceptions, but you can say perceptions because bacteria do perceive their environment and respond to it.
Never mind.
And so the hand of Darwin is on all that lives, yes. But there's a level of cognition that we have that, at least I think, they don't.
Yeah, we think we have free will, and we do certainly to some extent, although the microbes govern an awful lot of what we do, I believe. But yeah, we make conscious choices, which is not something that the natural world is particularly good at.
Yeah. Although some days I wonder if we really are making conscious choices, but that's another story entirely. You know what's interesting, Joe, is that you do fabulous science and you do fabulous outreach. And here you actually get into issues of, for lack of a better term, social structure and how to create better science, certainly. And all of these things are wonderful, and you're to be lauded for them. But as we start to wrap up, I wanted to have maybe one story from your time in the Obama White House. Can you share one?
Well, I came to visit you at University of Puget Sound years ago when I was in the White House, and I met with students and I met with you, and I visited your office, and you showed me your I visited your office where you showed me your stuffed microbe collection, and I see you have Ralph behind you there over your shoulder. And I was quite taken with the really tiny little ones because I realized that I couldn't put Ralph, that big streptococcus, in my pocket, but I could put a tiny Ralph in my pocket. And I ended up actually, you sent me a group of stuffed microbes, and I thought let's see, and soon after that, the president made a comment about flesh-eating bacteria, and he gave me a great opening to teach him some microbiology. So I put the little mini Ralph, the Streptococcus, which is the flesh-eating bacterium, in my pocket. You're not supposed to give anything to the president in the White House. And I gave him a little tiny Ralph one day, and he was very interested in it. And I explained to him what the flesh eating bacteria do. And what he really liked was, and you can see it over your shoulder there, was the fork and knife sewn on Ralph's chest. And he thought that was just hilarious. I heard him talking about that to a group of scientists years later. He described the fork and knife on the flesh eating bacterium. And so that became a tradition with the president to give him stuffed microbes every time I saw him. And it gave me a great opportunity, no matter what I was meeting with him about, I always got to talk at least a little bit of microbiology, and I think he got a great kick out of it.
I think that that is just a wonderful story. Can you imagine being surrounded by a whole group of people whose whole job is to keep you at a distance from other people, which is what that is, and you actually were able to bring a little bit of microbiology in there, which is wonderful. Joe, I've had the best time chatting with you today, and I really appreciate your time. And I want to thank you again, not only for the research you've done, but the friendship and mentorship that you've shown me. Thank you for coming on the show. And before we go, there is one thing that I would like you to kind of tell the listeners and viewers. There is one event in your past that made you know you wanted to be a microbiologist, and I see it in a photograph of yours.
Oh, yeah, it's quite memorable. In seventh grade, we had the great opportunity to look through microscopes at paramecia, and I was completely taken with that. And I never wanted to tear myself away from a microscope. And so I saved up my babysitting money for 6 months, and I bought this very old 1939 brass and black version that you can see here of a German hospital microscope. And it was in a warehouse of discarded medical equipment. And so I got it very cheaply. And as you can see, I still have it in my office today because I really love that old Lights microscope. And it's still a good microscope. It has such beautiful optics. Even then, Lights was making great optics. And I used it for many years to look at everything I could find around me, from pond water to crystals of vitamin C. And so it was a pretty important moment in my history, and I will never forget that.
And that's always interesting to think about. Joe, thanks again for your time today. I wish you and your lab group and your lovely husband the very, very, very best. Thank you again for being on this podcast.
Thank you, Mark, and thank you for your passion for the microbes. We all appreciate that.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episodes, with wondrous links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Joe Handelsman is at the University of Wisconsin-Madison and is director of the Wisconsin Institute for Discovery. Many thanks to David Renata for superb editing as always, and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.